Hi, everyone. Thank you for joining us today. We are very excited and honored to have our guest speaker today. I'm Vasiliki Annis, Business Director of the Rosalie and Harold Ray Brown Center for Cancer Drug Development. Our mission here is to accelerate promising oncology therapeutics into the clinic through collaborative drug development. For those that may be new, we have a broad scope here in through the indication area of oncology and different modalities, stages, and therapeutic areas under that umbrella. I'm going to go quickly here as we have a lot to go through, but please don't hesitate to contact me or my colleagues at the MESH Academy, and we're going to open the conversation into what your research needs are and how you can engage with the center. We've had an exciting seminar series this year, launched with our external advisory committee members, which Dr. Scherer is also a member of our external advisory committee at the Center for Cancer Drug Development. And future seminars are shown here, and you'll get a lot of notices throughout the year. All right, so it's my pleasure and honor to introduce Dr. Stefan Scherer, CEO of 3T Biosciences. And as I mentioned, we are honored to have his participation as an EAC member as well. Dr. Scherer is a preeminent thought leader in the field of precision medicine and drug development and an expert in academic alliances and partnerships. He brings extensive experience in drug development, clinical science, translational medicine, and overseeing development strategies from early to late stage assets. He has served as global head of experimental medicine at GSK, chief marketing officer and senior vice president of clinical development at Selectus, a leader in clinical, academic, and public-private partnerships, where he served as the chair of the strategic governing group of the Innovative Medicines Initiative. That was an EU public-private commission to facilitate open collaboration and research. He has also served at Novartis as vice president and head of early development and medical innovation, vice president of global head correlates services, and he's led strategic development of biomarker programs across all oncology indications at Genentech and Roche Pharmaceuticals. So he's left no corner unturned in this industry at all. It's very impressive, and he's earned his MD from the University of Freiburg in Germany, and has graduated with honors magna cum laude, and also completed his PhD at the Julius Maximilius University of Würzburg, and he graduated summa cum laude there as well. And he had a postgraduate education and a lot of experience in members of boards such as MacuCell, a research-based skincare company, which provides regenerative medicine biotech solutions. So you can see that we have a huge honor to have him participate at the CCDD and have him here today to share with us his expertise and insights in drug discovery and development of biologics for oncology unmet needs. Stefan, we're so happy to have you here. Thank you so much. I will stop my share. And the floor is yours.
Yeah, thank you so much for this really nice introduction. And I'm deeply impressed what you know about me. So this is, that's a lot and a bit scary, but nice. Thank you so much for this really nice introduction. And yeah, I was asked to give a little bit of perspective on drug development and drug discovery for biologics, especially for oncology unmet medical needs. And I mean, we can skip that. So this was basically a little bit my introduction, but you have heard that. So in interest of time, I think when we want to develop a drug, we always need to think about the environment, in which kind of circumstances we want to develop the drug. And I mean, if even despite all the efforts, millions, billions, trillions probably, which have been put into cancer research, globally, still one patient dies every 4 seconds. And I think there's certainly some sobering news. On the other side, there is hope. There is basically more than 4,500 compounds in preclinical development. There is also basically a lot of work ongoing in internal of big pharma, big pharmaceutical corporations, but the majority of the innovation actually comes from biotech. 60% of the innovation in big pharma or in healthcare industry actually is driven basically from academic or biotech. On the other side, basically, at the moment, there's roughly speaking 500 molecular targets in the clinic. 46 of those basically are first-in-class pipeline compounds, meaning breaking grounds, we're going into new biology, we're going into new targets, we're going basically understanding the disease certainly better. On the other side, I mean, this everything needs to be paid for, and the costs for innovation, the costs for healthcare, especially in US, is astronomical. So with 150 billion in healthcare costs, and there has been also significant drug launches, so to say, in the last 10 years, so more than 150 overall. And I think when we look into that, and especially then basically in the what I call situational context, so in 2022, in US, it's estimated that roughly speaking 1.9 million new cancer cases will be detected, and around 600 of those will die. That means we do see 5,200 new cases per day, and we also see, roughly speaking, 1,700 deaths every day. And on the other side, basically in cancer development and in cancer research, over the past 50 years, the outcome for cancer patients have significantly improved. So in the 70s, 80s, basically, roughly speaking, 50% of the patients overall basically were alive after 5 years. Nowadays is about 70%. So this is certainly a huge improvement in general. However, there is still diseases, and this brings me basically to the unmet medical needs here directly. Pancreatic cancer, glioblastoma, GBM, non-small cell lung cancer, and others will certainly remain below the 50% mark. And there is more of those. When this is kind of what I call basically, in essence, Oncology 3.0. The reason why I call this Oncology 3.0 is actually, if you look into the decade from 2000 to 2010, I think we can clearly say this has been the decade of genomics. So the HER2 amplified breast cancer has been detected, BCR-ABL, the BRAFs, the EGFRs, and others. In the 10 to 20s, so to say, we had the decade of immuno-oncology, a little bit still the decade, so to say, of the angiogenesis, or let's say modifications of the tumor microenvironment with the VEGFs, the PD-1s, PD-L1s, and others. I think now we are kind of living and getting into the wave of area of integrated data, which means we understand cancer better, but also the data we have to manage become more and more complex. So we can do ctDNA, we can do NGS, we can do whole exome sequencing, we can do genomic profiling, we can do a lot. But I mean, in order to bring that into context, this is an enormous amount of data. This is an enormous amount of things to understand. And this is beyond basically an Excel sheet. And so we need really integrated systems in order to understand what we're dealing with, and or better to say, how we can do this. The field is emerging. So this means basically the innovation and the product life cycle is shortened. So 10 years ago, basically a product on the market who has been basically appreciated in the cancer and cancer community has been around 7 to 10 years. Nowadays, we are down to 2 to 5. So meaning the innovation cycles become much faster. The innovation cycles become also much more frequent. And we also know that we target now a much smaller patient population and patient subgroups. And we also basically target different ways how we treat cancer. Before in the genomic medicines, we targeted mutations, we target the overexpressions. Now we are basically looking into immune therapies. We're looking into modifying of the tumor microenvironment, and so on and so forth. The other part, basically, when we look, and this is taken from a McKinsey report, actually, which is a really good summary in my mind. So when you look kind of where is really a persistently high unmet medical need, I mean, there's many parts. I mean, it's done basically by patient size versus unmet medical need, low to high. Certainly, and high unmet medical need is in the non-ALK, non-EGFR driven non-small cell lung cancers. Colorectal is still something which we need a lot of improvement. I mean, in third line colorectal, the data are not looking really good, as well as there is not too many drugs approved, and the ones we are approved have a very, very short PFS. In the world, basically, of HER2 positive, HR positive, HER2 negative breast cancer, there is still an unmet medical need, HER2 HR negative as well. Melanoma is basically a little bit in the middle. You have seen there has been a lot of progress in the last couple of years with 6, 7, 8 lines of standard treatment nowadays. On the other part, basically, when we target a disease like cancer, the fundamentals of cancer are certainly important to take into consideration when we look for drug and drug development. So one, of course, is heterogeneity. And I think we learned early on, or kind of 10 years and a little bit more, we learned that intra-lesion heterogeneity is real. Meaning if we punch basically into the upper left corner here or in the lower right, and would take a biopsy, we would certainly get to different results. We would eventually even treat the patient differently because the mutational spectrum could really be different. Other than that is also that if you biopsy a lesion, metastases versus the primary, we may not also have the same outcome and may not have the same result. A very important lesson basically is the temporal heterogeneity, meaning a tumor today and a tumor in 6, 8, 9, 10 months is not the same by the mutational background. So in other words, basically, if you want to treat a tumor, we cannot necessarily rely on a sample taken 2 years ago and believe that the tumor is still the same, or the mutational spectrum landscape, as therefore basically the context in which we want to treat the tumor is still the same. So the patient selection in the 2000s, so to say, has been relatively straightforward. What I mean with that, we looked at 2 things mainly. We looked at the tumor cell, basically cell surface, overexpression of HER2 and others, mutational KRAS, EGFRs, BRAFs, and others, basically intracellular. I think nowadays we understood that the tumor microenvironment is equally, if not more important, in order to identify patients and the right patients for the right drug, meaning the tumor microenvironment is also not a homogeneous compartment, it's heterogeneous. So we have what Google would probably call a 10x problem. So you have the heterogeneity of the tumor, and the heterogeneity of the tumor is actually overlaid by the heterogeneity of the microenvironment, because you have stromal cells, you have infiltrating immune cells, you have endothelial cells, it's vascularized, non-vascularized, you have a tumor microenvironment, you have TILs, TAMs, etc. So when we take all this into account to characterize the tumor in the patient by just taking a biopsy and understanding a mutational spectrum alone is certainly not helpful. The other part, basically, when we look into how we want to develop drugs and how we want to apply the drugs, basically then from pharmaceutical development perspective to the patient, most of the things what we have done in the past have been very much looking into a kind of a PFS, progression-free survival situation based on clinical parameters, meaning you had here on the right hand, you had basically curative surgery, the tumor volume, the tumor basically burden went down. And if everything went well, the tumor stayed down for quite some time. But then at one point in time, basically this tumor started to grow, but until we could see it and could detect it with any kind of radiographic method, ultrasound, CT, MRI, whatever, there was some time. But nowadays, when we take ctDNA, for example, which is a lot more sensitive and gives us a much earlier and a much deeper insight what's going on, we can actually detect the tumor up to 6 to 12 months earlier prior to the relapse we can see on a radiographic thing. So the time of intervention is in the future moving to this point away from that point. So we will start to treat patients more on a molecular relapse rather than we will start treating patients basically only on a radiographic relapse. And I know many patients are kind of really investigated with ctDNA on a routine basis. And Professor Lenz here is a very big proponent of it and takes ctDNA very much into treatment considerations already now. I give you an example, just a quick run through how important that is. So this was a study I ran actually during my time in Novartis, and this was basically an PI3 kinase inhibitor. It was a very classical study, basically a one-to-one design, patient, roughly speaking, 1,150 patients in the HR positive, HER2 negative, locally advanced metastatic breast cancer, regular endpoints, PFS, etc. It was basically buparlisib, which is a PI3 kinase inhibitor, plus fulvestrant versus basically placebo plus fulvestrant. When you look at the outcome of this study, what you see here, basically, it has actually been statistically significant, but not clinically meaningful. So the PFS difference here, what you see basically from roughly 2 months, is not something which is clinically meaningful. It's not something basically where you would really put a lot of hope on for your patient and or maybe even prescribe so as a drug to the patient. And this was done basically as an all-comer situation, which was at that point in time supported by the company. When you looked into the patients who were basically on a tissue analysis, only on the tissue, looking at the tumor samples. And the tumor samples have been, you could take any tumor sample, 6, 7 years old, 2 years old, 3 months old, it didn't matter. When you looked at the patients who have been PI3 kinase positive in the tumor samples, you could see the delta was widened, but the statistically significant was basically fading away. When you then basically looked into patients who had, on the other hand, on the ctDNA level, positive PI3 kinase, you could actually see what you want to see. You could actually see what you want to see from a targeted drug. Number one, there was a much bigger delta between a PI3 kinase inhibitor and placebo. And in the PI3 kinase negative population, you actually see there's absolutely zero benefit, which makes sense because there's no PI3 kinase, there's no target basically for the drug. There shouldn't be any benefit. So that tells you basically, if we assess the tumor on a much more accurate way when we assess it at the time of diagnosis, and not basically with samples which are have a lot of years, so to say, spent in the banks, and we don't know if the tumor is still the same. So there is really an example why a dynamic assessment is really necessary. That's another study we did with Nick Turner from Royal Marsden in London. This was a neoadjuvant study, basically also on a PI3 kinase inhibitor. So we took basically at baseline ctDNA, we saw that for this lady had 12 copies, post-surgery, post-neoadjuvant therapy, 0 copies. Basically, this lady was disease-free 2 years later. Another lady, similar thing, 16 copies, so to say, at baseline, 7 copies remained after neoadjuvant therapy, meaning the neoadjuvant therapy and surgery hasn't been too successful for this lady, unfortunately. So she had relapse, we can call it, with a ctDNA storm of about 200 copies after 6 months later. So at that time point, we knew already this will happen. And that's something which you need to take into drug development, because this can also be your surrogate endpoint. And this is where the field is moving. They're using also ctDNA as a surrogate endpoint and not only as a monitoring tool anymore. Moving on. So when you look really kind of in a way how the drug development is done and how it works, I mean, this is not much new to you. Just basically illustrative. There is really a lot of molecules basically in the beginning, but when you go through phase 1, phase 2, phase 3, and in a phase 1, you basically have a patient population with escalation and doses basically, and you have around 20 to 100 patients roughly in a phase 1. Depends how much you accelerate. You have hundreds basically in a phase 2, and you basically have normally around on the upper hundreds to thousands in a phase 3, and then you come basically to an approval of that, and that of course takes a lot of time. So what do we need to do when we look into drug development and we want to beat the standard of care? So certainly, and also for registrational purposes, we need to understand efficacy, we need to understand safety, we need also basically patient convenience, and of course, I mean, not trivial. The financial aspect is certainly something which needs to be taken in consideration as well. So what are the standard primary endpoints? Of course, PFS and OS, but the difference basically we can also use what the difference makes in a secondary endpoint, meaning the percentage rate at a survival of 1 year or basically 2 years, what people call also a landmark analysis. The duration of the response is basically a patient kind of longer responding than the other, and also the duration of response following a kind of a complete response, so to say, how long is a patient disease-free at the end. And of course, safety, basically, how many grade 3, grade 4 adverse events do this drug really introduce? Is it basically not treatable because of some hematological toxicities, or quality of life issues, basically, which is also something which needs to be very much taken into account. The other part is also patient convenience. So is it something which the patient is, for example, intratumoral injections is not a route of application, which is the easiest compared to maybe taking a pill. The other part of the coin is really what I call cornerstones of success of drug development. And I think there's certainly three. There's the biology, there's the medicine, and there's the healthcare systems. So if those come together and merge, so to say, you kind of have a marketable drug. On the other hand, basically, if there is one disjoint from it, meaning the healthcare systems, the biology and the medicine meets basically, but the healthcare system, there is an issue, means the targets needs to show that there is still positive risk-benefit ratio. As I said, a 2 months PFS is not something which a healthcare system would support, even it is biologically active, and it certainly hits a need in the medical part. The other part is if you have kind of healthcare systems and the medicine matching, so to say, but you can't get to the biology, is kind of these undruggable targets. And we know many of them, P53, KRAS, and others with high unmet medical need. And the other part is basically if the medicine is not tolerable, for example, you have toxicity profile is not favorable, the route of administration is not patient friendly. Those types of things are also a hindrance in order to really develop a drug successfully. So here's basically some criteria and what you would really do when you want to start a drug development. And this is something which is kind of more generic, but I mean, this is really criteria we use if we start the development of a drug and or have identified a target on the biology, clinical, and of course on the healthcare system side as well. So is a target necessary and sufficient for the disease? Meaning if it is not necessary, you clearly stop or you stop early. It's not sufficient, meaning it drives some benefit, but not enough. Can we rescue it by using combos? Can we really combine it with something basically which makes the drug more efficacious and or the duration of therapy basically makes it longer? Yeah, BRAF and MEK, for example, is something here. If you don't know, then we need to do more studies. We need to understand that better before we move really forward. And is a target needed for disease initiation, meaning is this a driver? Is it also something which leads to progression and or basically maintenance? So ideally, of course, you would love to have all three, but if needed only for initiation, but no longer for progression, that tells you something in which kind of patient segment you can really develop the drug and in which patient segment you may or may not be successful. On the clinical side, what is the unmet medical need? So what exactly is this disease stage you want to go? And is there basically tools that you can really prove and measure that it hits the target and you have basically a pharmacodynamic response in the clinic? On the healthcare system, what's your benefit? If you come with a benefit which is basically 3 to 6 months, it's questionable. And the FDA certainly is not looking into statistical superiority anymore. It's really looking into, and you have heard that and seen this basically, is it really clinically meaningful? Does it have something which the clinic and or patients would appreciate, and doctors, of course, as well. And then the other part is, of course, also, I mean, if you want to do it, do we need the 7th, 8th, 9th PD-1 inhibitor? I think we don't. And I mean, and that's something where also the question is, if you have 4 or 5 drugs ahead of you in the market, you cannot be first in class. Can you be best in class? If you cannot be first in class and you cannot be best in class, why you develop it? So those are the criteria basically which we kind of constantly think through when we do a drug development. So drug development, especially for biologics, more than it is for kind of small molecules, has actually moved into much earlier risk assessments. So we really try ex vivo, so to say, if you want. We really try to identify liabilities as early as possible, and I will give you a few examples. So we do in silico tox for things like TCR-T compounds, by bispecifics, TCR mimetics, etc., etc. You're not going to do any kind of non-human primate studies or monkey studies or something like that, because it doesn't work with the immune system. It's totally different. So anything you would read out of those things is not really liable. So what you do is actually go into organoids, and mainly patient-derived organoids and not reconstituted ones. in order to really understand, so to say, what's my liability. You need to characterize it qualitatively and quantitatively, basically, and you need to know what's my threshold, what can I tolerate in order to go into the clinic, And of course, basically, you need to also describe the exposure in order to understand that. So it means your assessment, your risk is really an integrated safety assessment. And this is something which you do nowadays more in model systems, where you do more in basically organoids and these types of tissue cultures or microorganospheres and things like that, which are out there, not so much the classical way. When you look basically the diversity and also basically the complexity of tumors and responses to drug, I mean, when you kind of do it a little bit, when you look back years, you have the evolution of therapies. So in 1880, basically, Sir William Osler basically said, the good physician treats the disease, the great physician treats the patient who has the disease. So I think there's really some truth to it. So and this is basically the evolution of the therapies. As you know, by 2015, 2016, the first immunotherapy has been approved, and we really moved away from chemotherapy and targeted therapies. When you look into the evolution of medicine, so at that time, basically 60s, 70s, 80s, this was really one size fits all. Nowadays, we are at precision medicine, and I think in the future, and the future is basically now 2021, 2022, we're coming to precision medicine 2.0, which means we're using a lot more out of the body assessment for drug responses with patient tumor materials. And we basically personalized medicine and precision dosing in one assay, which we call 2-in-1. So we want to understand basically what is the dose as well as basically what is the drug we use for this patient, and this can be done. So and this is really an integrated way. You would really look basically into tissue cultures. You would treat the tissue cultures. Basically, you would look into the histology of the tumor. You look basically in the sequencing. You look basically in copy number alterations, expression profiles, etc. You expose this basically the organoids or microorganospheres or whatever you take to your drug. You see basically when and what is responding and what not, and that's basically what you then would apply, and you would then kind of treat the patient with. And this is, of course, a lot more complex if we talk about biologics than we talk about small molecules, and because the system in play with immunotherapies and biologic is a lot more difficult than basically just a genetic target. So good biology does not mean it will be a good drug. And I mean, this is also something which is comes through as you, I mean, one example is basically RAS and targeting RAS, so to say, which is basically prominent in the majority of cancers, and in some cancer types, basically, for example, pancreatic, it's actually a majority, with 80 to 90% of the cancers. So this has been discovered in the 60s, and we still struggle, so to say, to have basically a great target and a great drug for these targets, so that the biology and the drug is not always matching. If you look into small molecules versus the biologics, it's very clear basically that the production and the liability you take, kind of the biologics, are really produced by living cell cultures. This is produced by a chemical process, basically a synthesis, like aspirin and many others, basically. This is very process development, and this is basically mostly process in development. So it's a lot easier to produce a generic than to produce a biosimilar, because the process is actually part of the drug, you know. This is very heterogeneous and a very complex structure, versus basically the small molecule is very defined and very well defined and easy to be characterized. Stability, of course, of the drug is also in the small molecules long months to years, and in biologic basically is not as stable, and it takes a little bit more time to establish that. A big part, and this has been also led to cancellation of drug and drug development, is immunogenicity. So there's not much, if at all, immunogenicity to small molecules, but immunogenicity in biologics is a big problem. And that's also something we try to assess ex vivo as much as possible. So the problem is really to understand the unknown, and to reduce the unknown and to manage that, basically. And that's basically a challenge which drug development now, so to say, faces. And drug development has really created a kind of a non-clinical toolbox, and this becomes really standard now more and more to understand these risks and to understand these safety lags. This is an example actually from a recent approval, and this is basically VEGF bispecific for wet AMD. And when you look kind of in a way when this has been started from a target assessment, when the target has been identified as especially Ang, and so in which diseases it plays, from a clinical lead, entry to humans, start of phase 3 to FDA approval, which was just end of the year, beginning of this year. So you can see this took a long time to get there, and the time we can really save in the future, and we will, is basically the clinical testing, because we will move this clinical testing a lot more to the forefront than it is now. So these 10 years may actually be shortened here in the preclinical part, in the GLP tox, and this perspective, there's not much you can shorten. Lead identification, lead optimization, maybe a little bit, but that's still a little bit the problem in drug development, and moving that to ex vivo, this will really help to get things faster to the market. And it has received accelerated approval, and I mean, this can be granted if we write meaningful therapeutic benefit over an existing therapy, but this is conditional. So this means you need post-marketing trials, and roughly speaking, 10% of these accelerated approval drugs will be withdrawn because they fail to confirm the benefit in larger trials than they have shown in a smaller situation. Yeah. Why is trial stopped? You may have seen it and you may have heard it. Immunity, for example, is a CAR T therapy. They had a dual PSMA specific and TGF-beta resistant CAR modified T cell, and they had two clinical trials. They had basically two clinical trials, and patients died because of basically neurotoxicity syndromes, and also kind of as a form of a macrophage activation syndrome. So with these two patient deaths, basically the drug is dead. So that means there is no way that this drug would be developed further and will be brought to the market. Same thing happened with the MAGE-A3 development program, because MAGE-A3 binds titin, and with the binding of titin, you get a lot of cardiotox, and also you get neurotox. And in the first trial, basically 2 out of 2 patients died. And in a follow-up trial, basically with a slightly modified version, 2 out of 9 patients died, especially also on this neurotox. And I mean, those are the things which could not be identified preclinically, especially also this macrophage activation syndrome and ICANS. But you see still these things you still see in clinics, which of course is a problem. So in my company, we looked actually at MAGE-A3 and the original compound basically, and what you could see, it really hits the target very specifically, but it has also a very, very good binding to titin. And this was the problem in the clinical trial. So we actually engineered the MAGE-A3 now, where we dialed out titin. So yeah, we kept the specificity, but we dialed out the titin, so that this will go into clinic next year. And we hope with this kind of reduced liability or excluded liability that we will have a good drug we can bring to the market. The other part, and this comes basically to the last part of the talk, is a little bit like what are targets for biologics and what is basically good targets for biologics. So certainly cell surface antigens are limiting the success of high potency modalities in solid tumors. When you look here, high unmet medical need, and when you look, 75% of high potency compounds are developed in liquid tumors. You can look at them here in the list, but less than 10% of all cancers have liquid tumors. So this means basically, if we can identify similar targets basically in solid tumors, would actually be a huge advantage and move the field. And what we also do basically, we develop TCRs, TCR-Ts, and also TCR mimics. So what we engineered is actually a system where we have organs and cell, and this is just illustrative, so to say, on one of the examples, where we really test in all of these parts, basically, what kind of toxicity we may or may not see. And that means basically we take cardiomyocytes, especially beating cardiomyocytes, in order to understand, is there anything, so to say, on cardiotox, vascular tox. We look also in lung chips, so to say, is there anything in terms of pneumocystis, immune reactions, etc., apoptosis, and other things. We look in intestinal organs or in organoids in order to understand that. We look in liver spheres for the same way, and of course also pancreatic islets and others. And this is really a tox assessment, and an assessment we do in all of the development, basically very stubbornly, one molecule after the other. Go through that because this reduces the liability, and we try to avoid, especially with this kind of high potency targets, we are developing like TCRTs or TCRMs to avoid or dial out toxicity as much as ever possible. When you look at the tumor specific intracellular antigens, basically you have kind of three parts: the neoantigens, viral antigens, or the cancer testis antigens, basically. And they can be private or shared, and the cancer testis antigens, this is basically what we target. They are normally not mutated, and they are also not presented on normal tissue. You only see them basically in the testis, and then you see them again in cancers, but in the normal tissue you don't see them. That means this is an ideal target because it's a target which comes up in the tumors, but you don't see it in normal tissue, meaning if you target the tumor, you would get a great efficacy. Reality is not the same. So what you see in these things is often they are associated with toxicity because unspecific binding, meaning the peptide HLA you have and you target, is often structurally and or from sequence similar to other proteins where you can really have this. One of those things which are very much advanced in the clinical development certainly is MAGE and PRAME. And there is also multi-tumor associated antigen targets in the repertoire. And as you can see, so they go very much into phase one, in phase one, phase two, and soon basically probably getting to the market. And so the field becomes, for these high potency targets, a lot more crowded. So this is certainly a therapy of the future, and the liabilities basically need to be seen. If you want to look, and it's a study we did here as well, if you want to look basically how big is your market, what tumor you want to target, which is something you want to go in, you look at RNA and you look at protein. So if you take RNA and you take protein, you kind of overestimate or not, and I really don't know the answer, the difference on the market size, as well as basically some tumor indications. You would not even see on a protein assessment, but you see it on an RNA assessment. So that means there is certainly a difference if you want to understand your target, what kind of modality you look into. I mean, here you can see, for example, this is MAGE-A4. So in uterine cancer, if you look at protein, it doesn't come up, and but if you go in RNA, you actually see it. And there's often really a big difference. The same basically happens here. For example, colorectal and PRAME, a similar situation. The other part basically is as I said, with these high potency compounds. And when you look into the toxicity, basically low versus high versus the potency, low versus high. Monoclonal antibodies, ADCs, modified ADCs, second generation ADCs, if you want, bispecifics, CD3 bispecifics, and CAR-Ts, you certainly have a higher part of toxicity, but your potency is also better. So what we did actually, we're building TCRTs and TCR mimics, and which are highly specific because we run it through a yeast display platform, and the yeast display platform has billions of proteins basically displayed. So with this kind of, and this is an AI/ML driven mechanism, which really helps to identify on tumor off tumor on target effects and side effects and toxicity. So that's something which we think we can really engineer out. So that means we can keep really the high potency, but we bring the toxicity down. So we believe we have molecules which have the potency basically of really the TCR mimics, TCRTs, highly specific, but from a toxicity level, basically comparable to an ADC or a second generation ADC, which makes it in the clinic a lot more tolerable and a lot more palatable. When you use this, I think we can really kind of the target MTD is here certainly displays the therapeutic window. And when you look into that, basically with the platform we have engineered and which is currently basically also others looking into similar ways to do this, it's really about to increase the therapeutic index, meaning we can dose either higher or we can dose longer. And the question is basically how do you hit the target, and basically how long do you hit the target? So to say, if you don't get your T cells into the tumor because you can't dose high enough, your compound is not effective. If you don't get T cell activation, even you bind T cells because it is not activating, that means also that your tumor is not affected by the therapy. And that means basically it's not efficacious and it's not beneficial. So with this method, we believe with an increase of the therapeutic index, we have certainly a high potency and a less toxic drug, which gives us the opportunity to leverage this larger and wider window. So this is something we looked into every single TCR we do. It's basically, you look into the peptide versus basically the CDR binding probability. So if you see kind of in a way, it binds very nicely, very high, but that's something you want. This is high potency. This is basically something you can hopefully expect. This has not much of side effects because it binds, but doesn't bind basically a lot. This one here is so-so. This is in the middle. This certainly binds other things, and this is the ugly part. So this is something where you would never go into clinic with a TCR like that. So you would really try to get the TCR in this range, and that basically needs a lot of engineering and also a lot of testing for this kind of bispecifics and biologic compounds. This is my last slide. So because I promised I will be done in 40 to 45 minutes. So and this is really what makes a good drug. So, and why, where do we have a go or no go decision with it, so to say. And I mean, this is certainly not the Bible of drug development. I mean, definitely not. But it's certainly a framework which we are kind of applying to many of the drugs, basically. And the idea is basically, do we have basically at least 20% RECIST responses in patients? Basically, do we see a good response? Do we have also, and we do always ctDNA, we do always ctDNA in order to get a very, very early readout in clinical trials. And we want at least 40% of patients with a reduction of the allelic frequency in ctDNA by 50% in order to see this drug, this dose is efficacious. The ctDNA, as I said in the beginning, that's why I plugged it in, is not affected by it. You're not getting a tumor reduction. You're not getting possibly a response. No on target of tumor toxicity, manageable CRS, no loss of efficacy due to immunogenicity. That's a big deal because if you get immunogenicity, you get a lot of side effects, you get a lot of CRS, and also basically some things, as I said, immunity, macrophage activation syndrome, etc., that can really kill the drug. The no-go is certainly the reverse. Basically, if you have basically more than 20% basically loss of efficacy due to immunogenicity, you would not go forward. If you have manageable unmanageable side effect profiles, you would not go forward. If your ctDNA doesn't change at all during the various dosing regimens, certainly there is not too much of efficacy to be expected. Less than 10% response rate. I mean, this also doesn't lead to clinical benefit at the end of the day. If you have a low response rate, you're not getting basically a 6 months or something like that benefit rate out. And of course, basically, if there is no efficacy across range of target expressing tumors, if you're not seeing it, if this happens in, let's say, it happens in breast cancer, but in lung cancer and in esophageal or whatever, the situation is different, then you can still go because you have an indication-specific drug. But if that doesn't happen in either one, then I think it's very clear that there's no way you would take this drug forward. So with that, I really thank you, and especially for the opportunity to talk here. I really enjoyed it, and I really much appreciate that. So, and open it up for questions, comments, or whatever you want to say.
Wonderful. Thank you so much, Stefan. That was an exciting presentation and just so much rich information. Like you said, open for questions. Heinz Joseph.
Yeah, thank you, Stefan. That was a great overview. I have a question, because with the new immunotherapeutic drugs and compounds, and we are combining them with known target agents, I wanted to know your take on what preclinical models should we use? Is there any priority? How do we need to watch out what models we should use? You know, there are these the organoids, and then there are the immunogenetic mouse models, and then there are the PDX models. And I think sometimes we are struggling. What is the right model to show? What is the next step to show proof of principle or early indication that this is a valuable drug? So I wanted to have your thoughts on it, Stefan. But it was a great presentation. I really loved it.
Oh, thank you so much. So what we try to do, and I mean, this is still a little bit early days, to be honest here. So what we really like to do is we look into organoid, or what people call this kind of microorganospheres, which are directly tumor derived. We're not looking basically in organoids which will be reconstituted by PBMCs, because what we identified is basically with this kind of reconstituted organoids, you're actually not getting much of a tumor infiltration and of the T cells. So especially for those compounds we develop, it hasn't been helpful. If you look at really derived organoids from patients directly and cultured, so to say, that is a richer information set we are getting, and we try to focus on those. I hope this works, because we don't have much of clinical data yet, but the information we have so far is it looks really promising if we stick to this type of model systems. Yeah.
Oh, Heinz Joseph, you're muted. Nope, he had a problem. Let me see if we can try to unmute.
Sorry about that.
Yeah, no worries.
This mute and unmute doesn't work very well for me. Yeah, I think you're absolutely correct. I think there are different opportunities, and I think they will probably change over the next years to come, depending on the mechanism of actions. And I think with the development of humanized models, I'm much more excited that this could be integrated in drug development, but they're very expensive and we have to have a learning curve. So I think I'm looking forward to all these new developments, because I think it will speed up. And with the CCDD, we are trying, as you know, with your help and Ivar's help and everybody who's involved, to have strategic alliances to make actually the next step faster and better and more efficient, not to lose too much time and money to get stuck at a certain level when the expertise of others could really accelerate the development.
No, absolutely. I mean, that's a, I mean, without being here on a marketing side or so, there's a few companies who really kind of came a bit more cutting edge out, you know, in these types of things. They also, I wouldn't say they're FDA approved, but they certainly basically talking to the FDA that we could use this basically for IND packages. And that is definitely an advantage in the drug development process. Yeah.
Great, thank you. Payam, I see you have a question in the chat. Would you like to ask directly, or you can read it? Are you?
I see it.
You see it, yeah. Okay.
Yeah. Yeah, I mean, for breast cancer, I think just for precision drug screening for breast cancer, I think, let me answer in twofold. I think breast cancer is one of those areas where I also think ctDNA, for example, plays a big role for different reasons. And if you take, for example, the HR positive, HER2 negative patient population, which is relatively big, and you take the elderly patients and 75 and older, if they all need to be treated, for example, with an AI or fulvestrant and whatever, or not, I think this is a question. So I think ctDNA will certainly help in order to escalate or de-escalate treatment here. What I mean with that is basically if I have a 75, 80-year-old lady who has this type of breast cancer, only CTDA negative, would you really want to give her 5 years of these drugs and with all the side effects? And the reason why I say that, this is a very acute problem basically I'm having, because my mom is one of those patients who has breast cancer and is above 80. So CTDA negative. So the debate is very much, should she take the drug or should she not take the drug? And the side effects on bone pain and these types of things with these types of drugs in this age are really significant. So I think that's going to be one part where certainly precision drug screening and development in breast cancer will play a role. That's more on the treatment side. On the development side, I think we need to really look into the subtypes of cancer. And I mean, as you know it very well, the mutational spectrum on breast cancer is not the most prominent one compared to other diseases. So I think the tumor microenvironment and these types of things will certainly play a role. And that's what I said, if we can really identify that in an ex vivo setting and basically then identify what is the best drug for this patient, I think that's where the field is moving in a very speedy way. Yeah, I hope this answers your question.
Okay, thanks. Any other questions? Anyone? I know you mentioned, oh, Camilo, sorry, I didn't see your hand.
I just raised it up. Thanks, Stefan. It was an amazing presentation, great overview. Thank you. I'm just wondering if you could elaborate a little bit on the regulatory challenges that biologics face, the typical challenge that they face compared to small molecules. If you could talk a little bit about, for example, more complex technologies that have both like ADCs that have an antibody and a drug together.
Yeah, that's an excellent question, because you kind of hit the nail on the head here with these things. Is basically we develop it, but what in the preclinical development process is okay for the FDA and or for regulatory bodies in order to approve it, and that's a struggle. Yeah, it really is, especially in the immuno-oncology field, because if you go into mouse models, if you go into even monkey studies or something like that, I mean, you essentially, let me be blunt, you can forget it. Yeah, anything like that is not translatable to humans. So what we have, what the FDA is really using, it's at the moment is this kind of organoid parts where they accept actually that as an IND enabling data package. So if you dose them and they are really derived from patients directly, fresh, and used directly, and then you can basically dose these organoids, so to say, with your drug and expose basically the drug to that and measure cytokine release on that. Basically, they actually accept that. And there has been a couple of workshops last year, and there's actually in the next few weeks, there's a paper coming out about that. I'm more than happy to send it to you, how this can be done. And this is basically talking about the peptidomes and which can be done on that ligandonomes, what they call it now, basically what kind of ligands you have to put on that. And that's what they want to take into these databases, as well as basically into these assessments when you use these drugs and bring them forward. Yeah. But they moving to the direction that you can actually run nowadays a drug development completely basically on animal free. Yeah, with that in this part. Yeah.
Great, thank you.
Great, thanks, Camilo. I think that's a fantastic way of closing out this session. We will have this video recorded. I know there's some questions about where these videos are. We'll send an email with links to the previous external advisory committee members who presented, and including Stefan's today and any additional seminars that we've hosted. So they're there for your viewing. And also for additional questions, please feel free to reach out to us and we can get these questions back to our EAC members if there's anything that comes up later on after you've been thinking about this for a while. So really appreciate everyone's time today, especially Dr. Scherer. Thank you so much. And
anytime.
look forward to seeing you very soon.
Yeah, I know. Looking forward to that.
Sounds good. See you in a couple of weeks. Thanks, everybody.
Okay. Thank you.
Take care.
Take care.